IRI, New Delhi. Sir has completed his graduation in agriculture in 1997 from Bidhan Chandra Krishi Vishwavidyalaya, Mohanpur, West Bengal. He did his MSc and PhD from Division of Plant Physiology, IRI, Pathology, IRI, and he was awarded awarded gold medal for both MSc and PhD. And in 2003, he joined as scientist in Central Research Institute for Jute and Allied Fibers, Barakpur. And in 2009, he joined as a senior scientist at National Bureau of Plant Genetic Resource, New Delhi. And now he's working as principal scientist at Division of Plant Pathology, IRI, and his research interest is on working on viral diseases of plant, and he has identified several strains of begomovirus, which are species which are infecting jute and allied fiber crops, pulse crop, vegetable, and mustard. He has also developed molecular diagnostic technologies for detection of various begomovirus strains. And with this, he has recently started working on genome editing of virus crops for development, to develop the strategies for their management. And sir has also gone through training by getting DBT fellowship for genome editing initiative at Washington State University, USA. And he is having nearly 48 renowned international publication. With this, he is also awarded by as a Young Scientist Award from Indian National Science Academy, Indian Science Congress Association, Indian Council of Agriculture Research, and National Academy of Agricultural Sciences. So now I would like to request you, sir, to take his lecture. Thank you, sir.
A very good afternoon to each and everybody. Welcome to this training program again, and this chilly, foggy winter, and after lunch it is very difficult to keep your eyes open. But don't get sleepy, because someone is watching. I need a pointer. So as I told that don't feel sleepy, someone is watching. Who is watching? A lion. Who is lion? What we know about lion? Lion is the king of the jungle, correct? But just think, can lion alone be the king of a jungle if the lion is not supported by a congenial ecosystem, starting from the grass to the initial feeder, then secondary feeder, and then lion. So if this system is not there, lion cannot be the king of the jungle. Similarly, in a scientific ecosystem, the big discoverer and discoveries will not come if there is no small observation of many scientists. But often, being a researcher, being a scientist, we forget the role or the contribution of those small discoveries, small contribution of many scientists who help a big scientist to discover a big thing. Okay? But today, what is happening? The scientists focus relentlessly on the future only. Once the fact is firmly established, the circuitous path that led to its discovery is seen as destruction. So nobody try to remember the small discoveries. Only we try to understand, we try to remember the big discoveries. So if I will ask you, can you tell few scientists in this CRISPR-Cas9 technology? Definitely, you will tell few names like Feng Zheng, like Doudna, like Charpenter, okay, but they came very late. Initially, the concept of CRISPR was not as a technology. Never, no big discoveries of any technology is developed by targeting that I have to develop a technology, and I start working on that. So, initially some young researchers mind try to find out some queries that how it is happening, and from there actually the entire science develop. So, today we will not go on this, that will just couldn't remember the small discovery, we will not do that thing. Rather, we will give a tribute to all the heroes of this technology by remembering their journey with the landmark discovery they made. And that is why today's class is the tribute, because for any, this is the Indian culture that before going to tell anything, we first give a tribute to all the seniors. For that only I am standing here giving a lecture, because if those discoveries were not made by those scientists, we are not here, you are not here as a trainee. So, first let me start with the birthplace of this concept. It was a very small place in the Spain, which is known as Port de Santa Pola. It is a very small place near the sea, and it is actually a very marshy land full of salt. You can see this kind of hillocks of salts deposited every year in this place. So, from this area actually the concept of CRISPR originates, and this was originated in the mind of a very young researcher in the late 80s, when he was around 25 years age. So, he is the person Francisco Mojica. So, he is the pioneer discoverer of the concept of CRISPR. So, he began his PhD work in 1989 at the University of Alicante at the port of Santa Pola. He was working on a Halophrex mediterraneani, an archaeal microbe with extreme salt tolerance isolated from the Santa Pola's marshes. His supervisor observed surprising things. In the initial days, they observed that at different salt concentration, the restriction profile of the bacterial genome is different, which should not be there, because we know DNA sequence does not change with any kind of environmental condition. But they observed that those bacteria which is isolated from those marshy land, the salt tolerant bacteria, because that time the restriction profiling is a big aspect. So, when they did restriction analysis of those bacteria at different salt concentration, they observed different restriction profile, which should not be there. So, that is the cue, that is the question, that why it is happening like that. So, his advisor gave him this problem to find out what is happening to this bacteria, that it is different, showing difference in the restriction profile at different salt concentration. So, what he did genome sequencing. That time, initial days of genome sequencing by manual method of Sanger sequencing, he did a genome sequence of a bacteria, and while analyzing the sequence, he observed a curious structure. He observed multiple copies of near perfect, roughly palindromic repeated sequence of 30 bases, which is separated by some spacer sequence which is roughly 36 bases. And those spacers did not resemble anything, and those repeats also do not match with any microbe, that time whatever is known. So, he got interested in that. He forget his original thesis ideas. It is the this is the genius thinks in different way. So, he forget that thing. He tried to find out why such kind of curious structure will be there in the bacterial genome, why these repeats will be there, continuous repeat sequence. So, it looks like this is from his paper I have taken, that these are the repeat sequence, always similar kind of repeat sequence is present, and this is separated by some other sequence which is known as spacer sequence. So, if you draw a cartoon of these, it looks like it is a repeat, then spacer, repeat, spacer, repeat, spacer. So, it is a area in the bacterial genome which is having lot of palindromic repeat sequence separated by spacers. So, he got interested on that and tried to find out that whether such kind of repeats is available any other bacteria or any things. So, he also observed another bacteria, halophilic archaea, that similar kind of repeats are also present and similar kind of structure, repeat followed by spacer. So, he started to think search on the literature, and in one literature by Ishino et al. In 1987 from Japan, they showed the similar kind of structural feature in E. coli, but without any structural similarity, and this paper also did not mention anything, any specific thing about that. So, he again got curious and he wrote up a paper reporting this new class of repeats. Then he completed his PhD anyway, he got a faculty position, and then just like us, when you will also join in a university or in any institute, initial days, you will not have any grant, you will not have any lab space, a sitting space, a computer, nothing. So, he is also faced with the similar situation which we everybody face. Okay? So, there is nothing. So, but he did not give up. He thought that okay, I have my personal laptop, I will search what is this strange repeats by only sitting in the computer using the net connection through the bioinformatics. I try to find out what these repeats are doing through bioinformatic analysis, and he dubbed this short repeat sequence as short regularly spaced repeats or SRSRs. And then by 2000, Mojica had found SRSR loci in 20 different microbes, including Microbacterium, Clostridium, and Yersinia. So, in different bacteria, similar kind of repeat spacer arrangement he observed, but he could not able to find out what is happening. So, in 2002, another group of workers, Janssen et al., they observed similar structural features in many other bacteria, and along with consultation with the Mojica, they gave the term, instead of SRSR, they gave the term clustered regularly interspaced palindromic repeats, because they are clustered in a location, they are interspaced by spacer, and they are palindromic repeats. So, from here the CRISPR term originates, and they observed that invariably in all the bacteria, a series of genes always present upstream of this CRISPR loci. If it is the CRISPR loci, then always in all these cases there are few genes which is present upstream of this CRISPR loci. So, they call them the CRISPR associated genes or Cas genes, but there is no clue that what is the role of these repeats or spacers. But as during that time a huge attention was given to the repeat sequence, the singles repeat sequence, the SSRs and other things. So, people try to find out what could be the function of these repeats. So, there are many scientists evolved different hypotheses. Somebody told that they may be involved in the gene regulation, they may be involved in replication partitioning, or they may be involved in DNA repair. But after experimentation, all these hypotheses proved wrong. Here again Mojica enters, he is the genius. He started thinking in different way. Everybody when start thinking about what is the role of the repeats, Mojica start to think about what is the function of spacer. Okay? So, what happened in June 2003, he observed that in one of the CRISPR loci from an E. coli strain, one of the spacers matched the sequence of a P1 bacteriophage that infected many E. coli strain. So, among many spacer sequence, only one spacer sequence he could able to find out a match with a bacteriophage P1. So, he got very much interested and he observed that the strain which carry this spacer surprisingly show resistant to P1 phage infection. It was very surprising thing that in a bacteria he is observing a small sequence of bacteriophage in the bacterial genome, and that strain which carries that bacteriophage portion, it is resistant to the bacteriophage. So, he got curious and he searched 4,500 spacer from different bacteria, because that time so many people have already reported so many spacers and repeat sequence. So, he collected all the spacer sequence reported by all the scientists, and he observed among this 4,500 spacer only 88 spacer sequence showing some similarity with some known sequence, and about 2/3 of this 88 matched with viruses or conjugative plasmids. But remaining so many, we do not know what is their function, what is their matching, no sequence match anyway. Only 88 spacer, and among them 2/3 of this 88 is showing similarity with some bacteriophage. Okay. So, he was very much enthusiastic. He hypothesized CRISPR is an adaptive immune system of bacteria, and this is the clue, this is the first thing that he got this hypothesis. If he would not have hypothesized this, the technology was not come up. So, he thought that CRISPR is an adaptive immune system of bacteria, but he did not have any experimentation, he did not have any evidence, but he wrote a paper. Based on the hypothesis only, he wrote a paper and he submitted the paper to Nature. Okay. So, what happened? Just like all of us we face, that when he submitted the paper to Nature, immediately it begins a 18-month odyssey of frustration. In November 2003, it was rejected from Nature. They told the key idea is known. So, nothing, all of we, whoever has published paper, you know that journal, from the journal only one line come, either it has no novelty or it has something like that, without no proper justification. Similarly, he also faced the similar problem. Why I am telling these things, that a big man also faced the similar situation which we also face. So, then sorry. So, then in January 2004, he submitted it to PNS. It was rejected from PNS, stating that there is no novelty. Then it was also rejected from Molecular Microbiology Journal and Nucleic Acid Research. Ultimately, he submitted to a very low grade journal, Journal of Molecular Evolution, and it also took 12 months to review the paper, and ultimately he could able to publish the paper in 2005. So, starting from 89, the concept which is brewing in his mind ultimately got a final shape in 2005. So much long time. So, do not get frustrated if you are not getting result. Okay. So, during that time 2005-2006, some group of people started to think about what is the role of the associated genes which is present upstream of the CRISPR array. So, people reported that Cas5, which is now known as Cas9, has two types of nuclease motif, HNH and RUVC. There are two nuclease motif. That means, this Cas9 is a nuclease which can cut nucleic acid, but surprisingly when they isolate that protein, put a DNA with this, nothing, no cutting, no cleavage. Though it is having a nuclease motif, they cannot cut the DNA. So, it is again a frustration. So, what is the role of CRISPR and Cas9 in bacteria? They really confer immunity. So, this kind of questions are always coming up one by one. So, the experimental evidence that CRISPR confers adaptive immunity came from the work of Philip Howarth. Now, remember that he is not working on CRISPR-Cas9. He is not at all showing any interest on CRISPR-Cas9. He was a microbiologist, got a position in a company, private company, Rhodia Food, which is now under DuPont. His main job was to find out the fudge resistant Streptococcus thermophilus. So, the Streptococcus thermophilus is a bacteria which is used in that company to make dairy product like cheese and other things. Okay. Now, in that company as they have to produce the cheese regularly, they have to maintain this bacteria free of bacteriophage infection. Otherwise, if the bacteria got infected by bacteriophage, the bacteria will not be active and they cannot produce the cheese. So, his job was to find out a strain which showed resistance against bacteriophage. Okay? So, he searched on the literature and come across with the paper of Mojica that there is a system, could be a system, CRISPR system, which can provide, which could be a new mechanism for imparting resistance against bacteriophage in the bacteria. So, he immediately contacted Mojica and he started experimentation on that, along with a new many of you may undergone some 6 months or 1 year training program with a company for an internship and such kind of thing. So, he is the person Bangarao. So, he joined with Howard to find out how we can find out the CRISPR is really confers immunity or not, because Mojica's paper is only hypothesis, no experimental evidence. So, their job is, first job is that whether really CRISPR can provide immunity. So, they did lot of mutational analysis in the CRISPR loci and the corresponding the bacteriophage genome, and ultimately they concluded that addition or deletion of spacer sequence from the bacteriophage make the bacteria resistant or sensitive to bacteriophage infection respectively. So, if you have a spacer sequence, you delete the spacer sequence from the bacteria, the spacer sequence is coming from a bacteriophage, then the bacteria will be susceptible. If you add a sequence from the bacteriophage as a spacer sequence, then the bacteria become resistant to that bacteriophage. So, that is the big conclusion, experimental evidence, that if you have a spacer sequence coming from a bacteriophage, then the bacteria become resistant to that bacteriophage. And they observed that immunity dependent on the precise DNA sequence match between the spacer which is present in the bacterial genome and the target sequence in the bacteriophage genome, which is known as protospacer. So, remember that in the bacteriophage genome, the sequence which is targeted by the bacteria to take it into its genome is known as protospacer sequence, and when it comes to the bacteria, it is known as spacer. So, the sequence is same. When it is in the bacteria, we call them as spacer. When it is in the bacteriophage, we call them as protospacer. Clear? So, when they observe these things, then the question is that who does this function of adding this new spacer in bacteria? In the laboratory, they clone a spacer sequence, put it in a bacteria, but in the natural condition, how bacteria acquire those spacer sequence from bacteriophage? Definitely something is there, some enzyme, something is there in the bacteria who can take out a portion of the bacteriophage genome, put it into the bacteria. Who does this function? This is the question. Then if spacer involved in immunity, then why Cas gene should be present? If spacer is everything, if delete spacer and adding spacer is giving you the immunity, then what is the role of Cas9, Cas proteins? That should not be there. So, these are the questions. After this experiment, automatically this comes to their mind. So, they simultaneously work on the proteins also. They did lot of mutational studies in those proteins, confirmed that the Cas7, which is presently known as Cas1, is involved in generating the new spacers and repeats, but that protein is not involved in immunity. And there is Cas5, now known as Cas9, which is necessary for phage resistance. So, Cas9 is the active component of the bacterial immune system. So, they wrote the paper in Science 2007, and this paper actually kickstart the CRISPR-Cas9 research. But this paper gives rise again some questions, that now there is two things, one is spacer region, one is Cas9, and if you delete spacer, bacteria become susceptible. If you add spacer, bacteria become resistant. If you delete Cas9, bacteria becomes susceptible. If you add Cas9, bacteria become resistant. So, whether both these components are equally important, whether there is any coordination between the spacer sequence and the Cas9. You just remember during 2007, nobody knows about CRISPR-Cas9, what we know today. That time how the science developed, I am telling that story, that how people come to one by one today's technology. So, this question comes, that is there any coordination between CRISPR and Cas9, and do CRISPR transcribed RNA undergo any modification, that if the CRISPR is there, if their RNA is produced, whether this RNA undergo any modification, or how this CRISPR RNA and the Cas proteins interact with each other. So, this work was initiated by John van der Oost. He was a biochemist. He was getting bored to doing the continuous restriction analysis. That time, one of the major research was identifying the restriction enzymes from different bacteria and show their effectiveness. So, he is doing the similar work regularly and he got bored. So, he was about to leave his job. He thought that this kind of monotonous work. So, suddenly when he read the paper of Howard, he got some new inspiration. We have some new enzymes which also doing like similarly cutting the DNA, the restriction enzyme like things. So, I should work on that new enzyme. It should be something novel thing. So, what he showed, he inserted a E. coli CRISPR system from one E. coli strain to another E. coli strain, the entire cascade of the CRISPR and the Cas9 thing, he take out from one E. coli strain to another E. coli strain which is do not have any such kind of things. And this transfer of that entire cassette from one E. coli to another E. coli which do not have is very essential for biochemically characterize the complex of these Cas proteins, because if one strain is not having Cas protein and another strain after incorporation is having, then you can very easily compare what is the function biochemistry of those genes. So, they observed that a complex of 5 Cas proteins is responsible for the maturation of the pre-CRISPR RNA to small CRISPR RNA, and the palindromic nature of the repeats would lead to secondary structure formation of the CRISPR RNA. So, what is actually I try to show you in the picture. So, this is the Cas locus and this is the CRISPR array we know till now. So, Cas locus produce several Cas proteins and CRISPR array produce a big RNA molecule. So, this Cas protein goes and chew up this CRISPR pre-CRISPR RNA into small CRISPR RNA, which is having a secondary structure due to the presence of the palindromic repeats. So, he thought that this CRISPR RNA is main culprit to do everything. So, he thought that if in the bacteria, if this kind of small CRISPR RNA is produced, can we artificially synthesize this kind of CRISPR RNA? Definitely this question will come. You just see how the logics of a research work comes. So, can artificial CRISPR RNA work, and what is the target of this CRISPR RNA, DNA or RNA? Whether this CRISPR target a DNA molecule or target RNA molecule. So, what he did, he reprogrammed the CRISPR array, created first artificial CRISPR array both in sense and antisense orientation. So, he created a CRISPR array to target 4 essential genes in lambda fudge. So, from the lambda fudge, he took out some special sequence, put it in sense orientation as well as antisense orientation, and then put it in a bacteria to see that whether the bacteria showed resistance against the lambda fudge or not. And to their surprise, they observed that only in the sense orientation the bacteria showing resistance against the fudge. And if it is sense orientation, that means it targets the DNA. Okay. But they could not able to directly prove that thing. So, that direct proving has come with the work of Marafini. He was also a PhD scholar at that time. So, his supervisor gave him a work that transfer a plasmid from a bacteria to another bacteria, which is very common. You isolate the plasmid, transform to another bacteria, it will go. But this PhD guy was 2 years working, he could not able to transfer the antibiotic resistant plasmid from Staphylococcus aureus to Staphylococcus epidermis. Repeatedly he is isolating the plasmid. When he tried to transform this bacteria, nothing is going. As a student, we think leave that thing, If it is not going, I will not do this work. But this is the gentleman who did not give up. He thought, I have to find out why it is not going, because protocol is very established. You have only transformed the bacteria with a plasmid, but it is not going, that means something is happening. So he asked his chairman, let me do sequencing of this plasmid and the bacteria where it is not going. I would like to do a whole genome sequencing. And after sequencing, he observed that in the plasmid there is a gene called Nick's gene or Nes gene, and there is a spacer sequence which is available in this bacteria and which actually interfere the transfer of this, because when this plasmid goes, due to the presence of this spacer sequence in that bacterial genome, it cut down the plasmid and there is nothing. So maybe plasmid going, but immediately it cuts, so nothing is observed. So really it is happening. So what next phase? He tried to disrupt both this spacer sequence as well as this Nes gene. The spacer sequence, that sequence, he disrupt it in the original plasmid, modify these things, mutate these things, and also in the bacterial genome he did mutation in the spacer sequence, and in both the cases he observed it abolished the interference. That means now the plasmid can enter. So it proves that definitely due to the presence of this spacer sequence, the plasmid was not going. When any of these things is mutated, the plasmid can go. So he can stop there itself, that okay, I got my job, I have transferred, but he did not stop here. He thought that if it is going and binding, means going and this kind of thing is happening, what is happening with the CRISPR RNA, because he wrote those earlier papers that CRISPR RNA is produced. So whether CRISPR RNA of this spacer sequence, the CRISPR RNA which is produced, they can bind with the Nes gene at the DNA level or the RNA of the Nes gene. He tried to find out that. So to know that thing, what very nice hypothesis he developed, he introduced a intronic sequence in the Nes gene of this plasmid. So in the Nes gene of the plasmid, he introduced a intronic sequence. So why he introduced? So if the CRISPR RNA targets RNA, then what will happen? After maturation of the mRNA from this Nick's gene, the mRNA which will produce, they will not have that intronic sequence. So the CRISPR RNA can bind with the mRNA and can inhibit. But if it is in the DNA, then due to the presence of the Nick's that intronic sequence, the recognition sequence in the Nick's gene will get disturbed. So the spacer, the CRRNA cannot go and bind. Okay? Simple experiment, simple hypothesis. So he put these two conditions, that if after putting the intronic sequence, if we able to see the abolition of function or not. If we observe that interference is abolished, that means it is present in DNA. If interference is not abolished, that means again we are not able to transfer, then the CRNA targets mRNA. So they observed, after their experimentation, they observed the interference is abolished, which clearly experimentally showed that CRISPR RNA goes and binds to the DNA, not in the RNA. So one by one step, how the science evolved. So again the question, that how it is interfering, and if everything we are talking about the CRISPR RNA, where is the role of then Cas9? Okay. So far, just a recap, so far what we have learned at this time of 2007-8, what was the knowledge that time? We know that CRISPR-Cas9 system is an unique adaptive immunity system which is having two components, CRISPR which is made up of repeat and spacer, and some Cas proteins. Second thing which we know, Cas9 protein is a nucleus, but it cannot cut the DNA alone. Third thing, Cas7 helps in recruiting the spacers, Cas cascades help in cutting the long pre-CRRNA into CRRNA, but we do not know the function of other Cas genes that time. CRISPR loci produces the CRRNA that targets DNA. So up to this we have this much of knowledge, but there are more questions. What are those questions? Who cut them? Whether Cas9, but we know that Cas9 alone cannot cut. and CRISPR, that also alone cannot cut. So, do CRISPR RNA has any role to activate Cas9 and then Cas9 will cut? Definitely this question will come. As per the literature, if you see, the question will automatically come that if Cas9 cannot cut alone and CRISPR has the target thing, automatically if you put this equation, you have to come to this hypothesis that definitely CRISPR RNA has some role in activation of the Cas9 so that now it can cut the DNA. And then how to prove that? Because if you have to prove that, you have to show that okay, Cas9 and CRISPR joins together and cut some DNA and you have the cut piece of the DNA, okay, cut piece of the DNA in the bacterial system. But there is no evidence. The bacteria which is resistant to a bacteriophage, you will not see any DNA molecule of that bacteriophage present in the bacterial cell. The system is so much effective that after cutting everything gone lost. Okay? So, there is no evidence to prove that whether CRISPR CRRNA activates Cas9 and cut the DNA. Then how to solve the problem? Here is the pioneering work of Sylvain Moineau, who demonstrated that Cas9 is guided by CRISPR RNA and creates double stranded break in DNA. So, he first demonstrated Cas9 is guided by CRISPR RNA and creates a double stranded break, and they accurately measure where the cutting takes place. They observe that Cas9 cuts the double stranded DNA 3 nucleotide upstream of a NGG sequence of the protospacer sequence. So, at the NGG sequence, which they termed it as protospacer adjacent motif. So, this protospacer adjacent motif is present at the downstream of the protospacer sequence, which is present in the bacteriophage. Okay? So, they observed that a protospacer adjacent motif whose sequence is NGG, any nucleotide followed by 2G, is the protospacer adjacent motif, and preceding to the protospacer adjacent motif 3 nucleotides preceding to that, which is present in the protospacer sequence, it cuts the double stranded DNA. Okay? So, today's exam, some question will come on this, that whether NGG is present upstream or downstream. So, I think after that this confusion will get clear. So, again the question comes that okay, CRISPR RNA guides the Cas9 to cut. Do CRISPR RNA directly interact with Cas9, or it also needs some other protein, other things to interact with Cas9? Definitely this question, because their experiment directly does not show that CRISPR RNA and this Cas9 directly in vitro they are interacting, they have not shown. So, this question definitely come that whether the CRISPR RNA directly target the Cas9 or not. So, here a completely unrelated work of one scientist from Germany came into light. She was Emmanuel Charpentier. She is working on the regulatory RNAs in the Streptococcus pyogenes, a bacteria, Streptococcus pyogenes. She is working on the regulatory RNA, how the different genes sequence is regulated, different protein production is regulated in that bacteria. She is working on that aspect. And she did sequence the transcriptome through next generation sequencing of that bacteria, and while analysis she comes to a very unique feature. What she observed, that in the small RNA sequence, we know the most abundantly present small RNA sequence in the any genome is the rRNA sequence, followed by tRNA sequence. And they observed after these two class of rRNA and tRNA, they observed another class of RNA which is abundantly present in that bacterial genome, and that sequence is surprisingly the complementary sequence of the CRISPR RNA. So, when she tried to search that where this RNA sequence is located in the genome, she got more surprised. She observed that here is the spacer sequence and repeat sequence, that is CRISPR array. Preceding to that there are Cas9 genes, and preceding to that there is a stretch of sequence which is complementary to this palindromic repeat. So, it is in the opposite orientation, and this sequence is present abundantly in the bacteria, and nobody knows that what could be the role. Till now we know that this Cas and CRISPR, so they are the component of this our adaptive immunity, but suddenly her work showed that there could be another component which is the complementary sequence of these palindromic repeats. So, it is the near perfect complementary to the CRISPR repeats. So, definitely the question comes, what is the role? So, their group showed that this new group of RNA was essential for processing of CRISPR RNA and thus requires for CRISPR function. So, they are termed them as trans-activating cRNA or TRA-RNA. So, this TRA-CRRNA or trans-activating cRNA is essentially required for maturation of the cRNA as well as for binding or guiding the cRNA to the Cas9. So, only the cRNA cannot go and bind with the Cas9, it requires the involvement of this tracer RNA. The tracer RNA is also essential for the Cas9 nuclease complex to clip the DNA. So, this work now again people try to revisit the concept. So, now there is 3 components of the CRISPR system, a CRISPR RNA, a Cas protein, and then TRA-CRRNA. So, 3 component in the CRISPR system. So, try to think whether we can do this CRISPR RNA and tracer RNA in vitro and in vitro isolated the Cas9 to reconstitute the entire process which is happening in the bacterial cell. Till now nobody has shown that artificially CRISPR RNA, tracer RNA, Cas9, put them together, add a DNA molecule, show that it is cutting in the appendop tube. Nobody has shown till now. So, she thought that we need to do this kind of things, then only we can definitely tell what is the function of each and individual component. But she was not having the knowledge of this kind of structural analysis and other things. So, she wrote some paper of a very famous structural biologist, the Jennifer Doudna. This lady is a very aggressive lady. Okay. So, she the Emmanuel Charpentier joins with Jennifer Doudna, a world renowned structural biologist and RNA expert, and they along with this Doudna and Charpentier, they produce the 2 RNAs, CRISPR RNA and tracer RNA, and they join them together to form a single guided RNA molecule or sgRNA molecule. They join the CRISPR RNA and the tracer RNA together to form only one RNA molecule, which is called single guided RNA molecule, because it is joined together. And then this concept of this sgRNA actually revolutionizes the CRISPR. So, what they did, they took this Cas9, express this Cas9 in the bacterial system, purify it, synthesize this sgRNA artificially targeting any gene of the anything, and then bind them together to form a ribonucleoprotein complex, add a target DNA from which that spacer sequence has been designed, and they observed that system under appendop tube can cut the DNA. So, this is the first time they showed, yes, now we can use them as a technology. Till now it was only a concept, only science, only knowledge. Now, when they could able to produce it in the in vitro condition, then they claim that this could be revolutionizing the process, and we can knock out any gene, we can edit any gene, we can do whatever we want. So, their group first shows that there could be a possibility, the potential to exploit the system for RNA programmable genome editing. So, they see their group only first give this concept that yes, this system we can use to precisely modify any gene. So, till now what is summarized, that in the system, I do not know, the Cas9 protein is not visible here. So, anyway, in the Cas9 protein, as I have told, there are two domains, HNase domain and RUVC domain, and this is the spacer sequence, followed by this green one is the tracer RNA scaffold. So, this entire thing is the gRNA and sgRNA, and this spacer sequence is actually the same sequence of this, which is called protospacer sequence. The spacer sequence is present in the bacteria, protospacer sequence is present in the fudge, but in the Eppendorf tube, this sequence is the target gene, and this spacer sequence you have to design from this sequence and join with this kind of scaffold. And then this is the protospacer adjacent motif. So, in the system what will happen, in the RUVC domain of the CRISPR now got activated, it cuts the protospacer sequence, and the non-complementary of the protospacer sequence is joined or is complementary with the spacer sequence, and then they get cut by the HNase domain. So, there was one question in the exam that which strand is cut by or in the Cas9 which domain is targeting the protospacer. So, the RUVC of the Cas9 targeting the protospacer sequence, and the HNase domain cuts the complementary of the protospacer sequence. The spacer sequence which we are providing is actually the same sequence of this, but remember that this should not, this spacer sequence should not have this NGG motif. NGG motif will be present only in the protospacer strand, not in the spacer sequence. So, NGG motif or protospacer adjacent motif is not a component of the guide RNA. The Cas9 will find out the protospacer sequence, and if it is finding a match with the protospacer region, then the complementary strand it will bind, and then the cutting will take place. Okay? So, when this entire thing was known, then we can summarize what is happening. What is happening? When a bacteriophage comes, then it gives its DNA to the bacterial system, and there are some Cas protein, they goes and bind with that bacteriophage, take out a portion of the DNA joined at the 5' end of the CRISPR array, and in that way the CRISPR array is increased, because it is acting as a memory for that bacteriophage that it has already come. So, what happens in the next phase? The Cas9 will produced, and then the CRISPR RNA is produced from the CRISPR array, and from the tracer it will produce the tracer RNA, and then when the next time the another bacteriophage will come, what will happen? From these three regions, the active Cas9 CRISPR RNA tracer RNA complex will form. They will go and bind to the complementary sense of the protospacer, and both the DNA strand, protospacer strand and the complementary protospacer strand will get cut by the activity of the HNH and RUVC domain of the Cas9, and then they got chewed up. And when they got chewed up, the rest of the things done by the other nucleases, exonucleases, endonucleases, so it will get completely chewed up. So, we will not get any kind of free nucleic acid available. So, this is the entire concept was demonstrated finally by Doudna and Charpentier. So, what actually these two ladies does? They joined these two molecules, CRISPR RNA and tracer RNA, by a small bonding, and they took this CRISPR RNA from a chromosomal DNA target. It is not from bacteria, any eukaryotic chromosomal target DNA. They create a CRISPR RNA, and then they join these two together to form the single guided RNA, and then they use this single guided RNA to see that whether the DNA get clipped, and they observe that DNA got clipped. And when they observe DNA got clipped, what is happening in the eukaryotic system? In the prokaryotic system, there is no DNA repair mechanism. So, in the prokaryotic system, in the Cas9 if it is there, they only cause complete disruption of the gene. There is no repair, no insertion, nothing. But in the eukaryotic system, as they have a repair mechanism, so this cut DNA ends, they can join by non-homologous end joining, and during that there is some nucleotide insertion or deletion which cause the mutation in the gene become non-functional. And if you have provided with a template flanking with this complementary sequence of these two regions, then this DNA can join together through homologous directed repair, and then there is a new insertion of a gene, new insertion of anything. So, this thing was demonstrated by the Charpenter and Doudna. So, again the same thing I am just avoiding. Now, this gentleman Feng Jiang, there is the biggest patent battle till now in the biological science between Doudna and Feng Jiang. So, Doudna only showed it could be a have potentiality to create genome editing. Okay? Feng Jiang showed that thing, yes, it is causing genome editing. Okay? So, it is a very interesting story in Feng Jiang's. So, Feng Jiang was attending a seminar. So, when he listened Doudna's paper, he immediately got idea. It was a seminar of 4 days. He came back to his hotel, okay, and he locked the room, put a sign, don't disturb, and started working on that, collaborating with his student in the lab, immediately designed that kind of CRRNA, Cas9 protein and all those kind of things. Within 4 days of time, he showed, yes, genome editing is possible in eukaryotic system in human being, and his paper came up in Science in 2013, and this paper initiated the era of CRISPR-Cas9 based genome editing. So, Doudna and Charpenter showed that it could be possible, and they showed, yes, it is possible, and that is why there is a biggest patent battle between Feng Jiang and Doudna. Who is the owner of this technology, Feng Jiang or Doudna? So, during 2013 to 2015, lot of companies jump into this system and they develop different vectors. So, this vector is having a Cas9 gene with a promoters, and this promoter is generally 35S promoter, which is a RNA Pol 2 promoter, and they also have a region where you have to design a 20 nucleotide spacer sequence from your target gene and just clone that small region under another promoter and flanking with a guide RNA stretch, which is conserved among all. So, only in this vector only you have to choose a 20 nucleotide sequence, finish. So, everything is commercially available, only you have to choose a 20 nucleotide sequence, put it in there. So, it will produce the inactive Cas9, it will produce the guide RNA, they will join together in vivo in the plant, and they can cause the editing of the gene. Okay? So, remember that this promoter is RNA Pol 2 promoter, but this promoter which is expressing the guide RNA is not Pol 2 promoter, it is Pol 3 promoter. That is why you cannot use 35S promoter, because 35S promoter is a Pol 2 promoter. The typical feature of Pol 2 promoter is adding a poly A tail and cap and the RNA. So, if you have a poly A tail and a cap in the gRNA, what will happen? The gRNA will immediately export it to the cytoplasm. Okay? So, genome editing will not take place. For the genome editing, the Cas9 and the gRNA should be localized in the nucleus. Okay? For the Cas9 in the bacterial system, they do not have any chromosome, they do not have any nucleus. So, for the bacterial Cas9, they do not have any nuclear localization signal which can drag that Cas9 protein to the nucleus. So, if you have to use a Cas9 protein from the bacterial system, first you have to add a nuclear localization signal in the 5' end of the Cas9 protein, so that that Cas9 protein will directly go to the nucleus, and you have to use a Pol 3 promoter for gRNA expression, so that the gRNA will synthesize in the nucleus and cannot come into the cytoplasm. So, when both these things is present, then only the genome editing will take place. Okay? So, this Pol 3 promoter like U6, U3, these are the Pol 3 promoter. So, this is the system. So, companies has developed for us. So, in that way, now it is coming up to a technology. So, what happens? The work started 20 years back. So, 20-year story only started in 12 cities in 9 countries. whatever I have talked, it was only 12 cities in 9 countries, but now within a year, investigators had reported use of CRISPR based genome editing in many organisms, including yeast, plant, nematode, fruit fly, zebrafish, mouse, monkey, human, everywhere. Now genome editing can be done, and you just see there are other techniques for genome editing like meganuclease, zinc finger and talent, but after 2014 when these commercial vectors are available, suddenly there is increase in the publication only based on CRISPR-Cas9 technology, and it is called the technology of the millennium. And it was written that I do not think there is any example of any field moving this fast. One report was came that per minute there are 9 papers are depositing or means submitted to different journals. Per minute 9 papers on CRISPR-Cas9. Many of them got rejected, but this is the way a technology has come up and everybody is now using this technology. So, main countries are USA, the European countries, China and Japan. There is no list of India till now, almost there is no report from India. So, recently we have published one paper in PLOS ONE. When I will discuss about my work, that time we will show you that what we have done. And there are so many patents in CRISPR-Cas9, so many patents have been filed. Can you see the name of Francisco Mojica here? No. So, that is the thing, the early heroes of CRISPR were not on a quest to edit the human genome or even to study human disease. When Mojica started working a curious structure, he did not think any idea that I have to develop a technology to edit human genome. That is the greatness of the genius. Their motivation were a mix of personal curiosity and their job demand, because their job, their PhD work, that thing. So, they are only trying to solve the puzzles, different questions arises, they try to solve, and ultimately solving all those questions, a knowledge become a technology. So, what Mojica is doing now? Mojica is not interested in genome editing, not at all. He is not in the run of any patent. He is now searching what are the other spacer sequence and what are their role, because 4,500 spacers and only 88 showing match. 2/3 of them is showing bacteriophage. What are other sequence? What are their role? Mojica is devoting his research now only on that aspect. So, students, this is the paper which I talked till now. So, you can read in the Cell, the heroes of CRISPR. It is freely available. You can download, very lucid writing. It is just a story format. You will enjoy the contribution of different scientists, how they contribute for development of this technology. Thank you. Any question? Discuss any question and
can you explain the concept of RUBC domain and this NEH domain?
there are different lectures for that. Here it is only the history part. Dr. R.C. Bhattacharya will take up the mechanism and all those things. I have just touched a little bit about the mechanism, but he will take it in a broader way, that what is their role and all those kind of mechanistic things. So, but in the related to the today's lecture, the history